For the next oral presentation is dual bispecific protein engager arm T cells targeting integrin alpha V beta 6 and PDL1 enhanced cytotoxicity against cholangiocarcinoma, presented by Settawut Buadee from Faculty of Medicine, Siriraj Hospital, Mahidol University.
Good morning, everyone. My name is Settawut Buadee, a fifth-year medical student from the Faculty of Medicine, Siriraj Hospital. Well, today is my pleasure to present my research project on the development of dual bispecific protein engager arm T cell, which I refer to dual BATs, as a novel immunotherapy strategy for cholangiocarcinoma or CCA. To begin, let me briefly introduce about CCA. CCA is a highly aggressive and lethal cancer emerging from our immune system. As you can see from the global incidence map on the right, Thailand has one of the highest incidence rate of CCA in the world. This underscores the major public health burden of CCA in our regions. Unfortunately, most CCA patients have very limited curative options. Even the current standard treatment remain ineffective and are associated with poor outcomes. This highlights urgent need for new therapeutic approaches. Recently, immunotherapy has emerged as one of the most important modalities that reshape cancer treatments. However, implementing immunotherapy in solid cancer still face several challenges, including tumor heterogeneity, tumor immunosuppressive microenvironments, and the risk of serious adverse effects. To address this issue, we have conducted this research. So I would like to explain the concept of our studies as shown in this slide. First, let me briefly introduce what bispecific protein engager or BiP is. You can think of a BiP as small proteins with two different hastes. One haste will bind to a tumor-specific antigen on cancer cell, and the other haste will bind to a receptor on T cell. By this mechanism, BiPs will bring T cell into close contact with cancer and redirect T cell to eliminate cancer more effectively. In our project, we have developed two specific BiPs. The first one is A20 CD3 BiPs. This BiPs will recognize integrin alpha V beta 6 on CCA, which is a promising antigen in CCA. And the other haste will bind to the CD3 on T cell. And the second one is the PD-L1 CD28 BiPs. This BiPs will bind to PD-L1 molecule, which is a negative immune regulators, and CD3 on T cell. We then isolated the primary human T cell and ex vivo armed them with these BiPs. When T cells are armed with BiPs, we call them BATs or BiPs armed T cell. After generating BATs, we will evaluate the functional activities with a specific method, which I will explain later. So in this project, we have three main objectives. The first one is to develop dual BATs by using two specific BiPs, which I mentioned earlier. And the second one is to investigate the integrin alpha V beta 6 and PD-1 expressions and their correlation with clinical particular features in CCA patients. And the last one is to evaluate the functional activities of dual BATs against CCA cell line. To achieve our objective, we design our experiment in 4 parts as shown in this slide. Now I will begin with part 1. First, we access our target antigens in the 44 Thai CCA patients using immunohistochemistry tissue microarrays. And we found that 54% of cases had high integrin alpha V beta 6 expressions, 47% are PD-1 positive, and 27% co-express both antigens. Moreover, these antigens and their co-expressions were significantly correlated with the lymph node metastasis, distant metastasis, and the lymphovascular invasions. Kaplan-Meier analysis in panel B, C, and D show poorness in overall survival in high expression group. In multivariate analysis, lymph node metastasis was the strongest independent risk factors. Finally, the flow cytometry and immunofluorescence imaging confirmed that KQ2138, which is a CCA cell line, was a strong double positive of our target antigens, while HeLa cell line was a double negative, so we used them in our experiments. let's move to the second part, which is the biopsy generation and characterizations. So in this slide, we show how we generate our biopsy using the lentiviral system to create a stable cell that secrete our biopsy. We confirm this process by looking at the IFE expressions and immunoblotting for tag proteins on biopsy molecules. We then evaluate the binding abilities of our biopsy. The flow cytometry panels confirm that our biopsy can effectively bind to its respective target antigen on both T cell and cancer cell. The fluorescent imaging in panel J demonstrates the clear immune synapse formation, which rare tumor cells are close contact with killing T cell. And the third and the fourth part are focused on the evaluate the functional activities of dual BATs. First, let's just take a look at third part, which is the anti-tumor activities. We use a two-dimensional co-culture system between the maturely expressing target cell and the full group of effector T cell. So, as shown in this slide, and we measure the mean fluorescent intensity or MFI at the 24 and 48 hours, and the cytotoxicity was calculated by the reduction in MFI compared with control. And here is the result. Figure A and B show a result in HeLa cell line, which is a double negative. You will see that all group of biopsy arm T cells show only minimal killings, demonstrating the high specificity and low of tumor killings. In contrast, figure C and D show a result in the KKU213A, which is a CCA cell line. The result is the dual bispecific protein engagers exhibit the highest cytotoxicity at both time points, while A20CD3 arm T cells show moderate but significant killing. Interestingly, PD-L1 CD3 arm T cell only show the minimal killings. In the fourth part, we evaluate the T cell activation, proliferation, and cytokine production. In figure A, B, C, and D, we detect and measure the late and early activation marker on T cell, which dual BATs exhibit the highest level of activation in both markers. And figure E and F show the T cell proliferations, which also dual BATs also demonstrate the highest, the strongest proliferation signal. And finally, the cytokine analysis, dual BATs markedly increase in the cytolytic molecules and polyphenol cytokines compared to other groups, as you can see from the results. In conclusion, our study showed that integrin alpha V beta 6 and PD-L1 are clinically relevant to CCA and represent a promising bioprognostic marker and therapeutic agents. And also, we also demonstrate that dual BATs or dual BATs arm T cell can markedly enhance the T cell mediated cytotoxicities and to improve the immunotherapy in solid tumor. Thank you.
From the analysis of the real clinical sample at the very first part of your presentation, it seemed like only 1/4 of the patients have co-expression here, 27%. But most of the patient sample have a single expression, right? So do you think how they're gonna benefit to your dual BAT?
Oh, you mean that like only 1/4 of patients show co-expression? So what is gonna benefit my biopsy, right?
Because in your in vitro assay as well, so
Yeah.
you test with HeLa and KKU line that
Yeah.
have dual, but it's also interesting whether your dual BAT how it's gonna behave if the tumor cell or tissue sample have only a single expression. Is it gonna benefit or it's gonna benefit only with double expression?
Actually, if we utilize the both biopsy as I explained, it will markedly enhance the T cell mediated cytotoxicity. But even the patient explains only integrin alpha V beta 6, I think it's not like it's not significant between the single A20 PD-L1, single A20 CD3 and dual BATs, because the second BIPs, the PD-L1 CD28 is augmented. The first one, if the patient only express the integrin alpha V beta 6, so like the efficacy might not be different.
So how you gonna suggest in real world applications of your dual BAT? So what kind, what is the primary screening, or what kind of patient should gonna benefit the most?
Currently, for the immunotherapy right now, the primary screening we base on the genetic testing or the immunohistochemistry, like the PD-L1. But in our promising antigen, the integrin is currently there's not existing established guideline that can screen. But I think it will benefit if we have the strong evidence that can screening the patient into the criteria to be benefit from our BIPs. So it would be whether the genetic testing or IHC. Thank you.